So, good morning. Thank you for coming. It's my pleasure to have here today Professor Marc Van Montagu. It's really a great honor that he could come for the opening of our PhD program in plant sciences, and also to give a lecture to all of you. So I would like to say some words about what Professor Marc has done during his research life. So Professor Marc holds a PhD in organic chemistry, in the biochemistry, and the bachelorate in chemistry from the Ghent University. Professor Marc, as most of you know, is a pioneer in the plant molecular biology. It's well known that together with Professor Schell, they discovered the TI plasmid, and is the inventor of the Agrobacterium transformation technology that is used worldwide to produce genetic engineered plants. Professor Marc used this technology to study regulation and discover of molecular basis of several plant physiological processes. He has major contributions to the identification of genes involved in the plant growth, development, flower, and also abiotic and biotic stress responses. Professor Marc has produced over 1,000 publications, and in the total of the scientific publications, he received more than 50,000 citations. What is really amazing. He won numerous prizes. He's a member of several scientific academic societies worldwide. For one of them is the Academy of Science from the United States. This is just one of the example. In the 2013, he was awarded the World Food Prize, what is really amazing achievement. And in 2014, he became Unido Goodwill ambassador for agro-business. Professor Marc was the founding member and the member of the board of directors of Belgian biotech companies, spin-offs from his lab. The companies were Plant Genetic Systems and Crop Design, that probably you have heard about them. These companies drive frontline innovations for biotech agriculture. Due to his accomplishments, he received in 1990 the title of Baron from the King of Belgium. But I find it really very nice achievement. Currently, he is president of the European Federation of Biotechnology and of the Public Research Regulations Initiative, and chairman of the Institute of Plant Biotechnology Outreach. And today, Professor Marc will tell us about plant sciences, discover, innovate, and communicate with the society. So thank you very much, Professor Marc, for being here today. It's really a big honor for me. Thank you. So the stage is yours.
Thank you, Nelson, for the kind introduction. And I think this is because you are recording it. It's not because you believe that my voice is already too low that it will not hear me. Okay, I had the luck and privilege of being now for 65 years following plants, not plants, science development, biochemical development. I thought maybe I can have a kind of title that looked a bit authoritative and didactic, but I think it helps. It helps for focusing the ideas on what is going on with plant sciences at the moment, and what is expected from us and all those who are or will be involved in plant science research. I think, your PhD program is also called Plants for Life, that all of us, if we look to nature, we are extremely worried by the damage that is done we didn't have to wait for the green movement to realize that, but it was good that people even made a political movement of it, because it's really extremely important that we see how can we preserve what is still left as nature, and what should we do, what can science contribute, what can science and technology contribute. Because what is going on, you know it, is the ever-increasing world population. After the Second World War, when I approached universities, then we were 2 billion. 50 years later we were 6 billion. Now we talk about 9.5, 10 billion. We know that what will happen, we will always go on clearing wild nature in benefit of the society. That is a very, very difficult situation. And that will be the essentials, and that's why I do it in that type of slogan way, that indeed we have to absolutely progress and as fast as possible with our discoveries, but we have to see that we use it, and that we use it in a good way, and that we can explain to society why we use it. So I want to symbolize plant science by this slide. We are all fascinated by systems biology. We want to know what in the living organisms, what are the chemical reactions that go on, how are they controlled, how to determine all the properties. But systems biology works well for yeast cells, and there's an enormous progress, but for more complex systems we are far, far away, and society wants applications. And that will only be there if we are not isolated in our ivory tower of the laboratory, if we have close contact and really collaborations and learn with interest what is traditionally done in agriculture, even in the history, what people were doing, what is the knowledge that is there, and how can we add the knowledge from one specialist to the knowledge of another specialist. Here I've seen you're in a good environment, you have, with your fundamental research, you are close to people who are to agriculture, but don't forget to talk to them and insist on the responsible organizers of your PhD program, that you have contact and that you learn also what is really agriculture, when it can be done, and the history of agriculture. Then you will really understand what the needs are and how the chances are that you can apply the science. Because with this picture, we also should think that we are not only talking about the science and the way science is done and the way we can apply it, but I also think it's important that we realize where the knowledge can be used in society. And we know there are enormous problems, because when people hear talking about science, they think that scientists don't respect society, they don't listen to society. And it's a fact. With rationalism, it's so easy to think the science, that's the truth, and the rest will follow, and all these people with their emotions and the society, they are used. And the study programs are given like that. You have no contact with social science, you don't have contact enough with economics, you don't understand what is a society, how people live, how you live, because all the stories about beliefs and not really viewing the society like it should be looked at. It's for everybody, for people who don't know science and people who know science. And it's very insulting to say, oh, you with your belief, because you have also your belief. It's logic. Science, you can only know rather well the basis of your own science. And even then, at each moment after 65 years, you know that it's only very few that we know what we should know, and we are very optimistic that in 10 years and 20 years we will so much know more, because we have seen it and that it happens there. But that's sufficient. For all the other sciences, well, we appreciate the science approach, but the facts themselves, we believe that the scientists, the way they work, they do the best, but we know opinions will change at a certain moment. But so we have we give mandates to them. Yes, go ahead, and we will believe in you. And the same in society. Indeed, if you see sometimes society in action, it's hard to believe in society. But okay, do that like you do in science. Ask questions and always challenge, and don't take things for granted, and you will progress a little bit. It's not a solution, but it's the only thing that we can do. Try to have knowledge about not only plants, but all aspects of science, or knowledge about our society, and then probably we can help each other. That's the introduction. Later we come back to that because if we start talking about that subject, it is for hours and days that we could talk. So, but it's just to keep your attention. Okay, the history of plant science brings us already on the way. We started in the 60s to get interested because molecular biology of prokaryotes gives us some cues already how these molecular macromolecules interact. Many of our colleagues went in mammalian science to solve cancer, to solve other problems in medicine. We thought, well, it will be cheaper if we could do these questions with plants, and we don't have to work with test animals, we just work with plants. And we got interested in what is this tumor that occurs and that was described since the beginning of the 20th century, and that gives proliferation, and all phenomena were described, and because you can have teratomes out of that, cells will start regenerating and new plants will fall. So it was like you had there tumor cells and cells that gives metastasis and gain development. So it was very intriguing. The history you can read in books where we have written it in Annual Review and others. But it was because we were in microbiology, we had the luck in the 60s to learn about plasmids, learn about insertion sequences, about movable elements, that we focused on plasmids, and technically we were able to isolate these very large plasmids because that was completely new. 200,000 base pair plasmids were exceptional, and we could show, and that's the most important that you remember, that it was a story about horizontal gene transfer. And we will regularly come back on that. We could show that it is a segment, a sequence of this plasmid that we call the tDNA that gets inserted in a plant cell, and then this plant cell proliferates and obtains new properties, and one of the new properties is making compounds that is not useful for the plants or for other bacteria, but was useful for Agrobacterium. That's a way of Agrobacterium to force plants, we can do it with many plants, to force them to make strange amino acids, a composition between keto acid and an amino acid that not other living organisms is using, and that only that bacteria can use. It's just a way in evolution how a plant finds new sources of carbon and nitrogen that the competitors will not use. But it was interesting that it was a DNA that is transferred and expressed in a plant cell, and it certainly gives new properties to this tissue. That's what they call horizontal gene transfer, and we will come back to that. Luckily, the science community in this, it was definitively proven and announced that you could use this as a vector for gene engineering in 1983, and we were actually very promoting this technology because since the mid-70s we believed that really that would make plant gene engineering possible. So we didn't see, is it the only case of horizontal gene transfer? Because we are convinced there are others. But anyway, we didn't have the tool to find other ways because technologies and sequencing didn't exist at that moment of the quality of mass spec. And there are many reasons that we could have focused on other subjects, but we wanted to see that we could use it for gene engineering. Because all the aspects of developmental biology are still there. Why this proliferation? Why this interference with cell cycle? And that still has to be. So the whole community followed and focused to use the tDNA for one hand on fundamental research, and it's symbolized by Arabidopsis. And thousands of scientists now use Arabidopsis as model system, and through gene insertion and rearrangements that can occur, we have learned a lot about plant development. And also, luckily, since the very beginning, there was work done for applied research that is symbolized here by cotton, because 70% of the cotton at the moment is transgenic in the world. So all your Eurobills are transgenic cotton. Don't be afraid of them, don't throw them away. They are not so dangerous. The way they developed by itself, we could talk about ours, but just for your remember that we started immediately to see that this fundamental research should be applied, but we had the luck that we had a minister of science. Well, in these days, education and science was the same ministry, who said, at the university, you don't make products. University, you are there for research. It's industry that makes products. And so in the States, we had seen what is a startup company. In the 70s, what is that? So we did it. And they gave us the possibility to make startup companies in Ghent, and that is the beginning of the application. But how the big industry then made that they make a monopoly of the GM technology for their production, and how the startup companies were always bought by the big industrial concerns. That's also a fascinating story where we could talk about ours that you have to analyze, and we will come back at a certain moment on that. But what you remember that when we did this startup company for applications, we had three successful, immediately approaches that have been applied also everywhere. That was the BT story that was done in Ghent. The first herbicide resistance for Basta was done in Ghent. And another technique that is not very much used, but has a lot of promises, but that were blocked by the major companies, that's hybrid vigor, because hybrid vigor is very important. It's only used for canola, could be used a lot for developing countries, but they have the patents and they decide to go ahead with it, although the story of the patterns, we can also come back to that. But for all three, there are bacterial genes that are expressed in plants. And I think there is also a reason for the complex of systems biology that these proteins interact a minimum with the developmental state of the plants, and that for that reason it's good to work with bacterial genes or even with synthetic genes if you work new properties in plants, because evolution is a very complicated molecular biology. So, after the transgenic work that was stalled for applications for nearly 20 years, it was all developed for around the three approaches that I have mentioned. We come back to that. There was, through the fundamental work, there was a lot of small interfering RNAs that were discovered, microRNAs, went progress to do what was called synthetic nucleases, and the whole science developed tremendously when the new tools were possible. And now we talk a lot on the new breeding techniques, and I hope that really they will bring substantial progress, that we have new applications that not only the big industries but all the small, medium companies will be able to do. Each school of agriculture who traditionally does the breeding, all the traditional plant breeders will be able to do it, and that is the hope with the new breeding techniques. But also it will be an enormous tool for the fundamental research. So when we saw an explosion of knowledge when we could make transgenics, it will be an addition to that, and together with the transgenics, we will know a lot. But already I want to ask your attention, especially for this lecture, to a publication in Cell from last year by Tom Check and John Argetzinger-Steins. It is a very nice article on showing that with the RNA world that develops, that you break the dogmas. And that is what science is, and that you never should forget. We always ask questions, we have the answers at the moment, but we go on, and with the new techniques we have new visions, and we see that our knowledge of the beginning is only part of a better knowledge that we have to have later, and that can be used. So we will not go in the beginning, enzymes are proteins, then came the ribozymes. RNA processing is at the end, but the trimming with exonucleases. But then we saw processing with introns, and we understood RNA editing. The splicing, it's to remove junk. That was accepted, and then suddenly you see, oh, alternative splicing is important, and so much information can be there. The ribosomal RNA for ribosomes, that's the scaffold for the protein. And then suddenly you see, no, this RNA has also a catalytic function. And each time for all what is happened, we will again, that article is worthwhile half an hour of talk to understand, but keep of it the concept, ask questions, and you will see that finally, when the answers are given, sometimes it takes 20, 30 years because the tools are not there to give the answers, but that's what science is and how science works. The whole methylation of DNA and the demethylation of DNA. In the beginning, it's something curious. What is it? And again, I give a review article. Each step in the plant growth and development is what we call now epigenetics is involved. There are genes involved that were not expressed and that then temporarily are expressed. Only at a certain stage they are useful, and in other stages they are switched off, because otherwise they would interfere with other processes. That's the way evolution works. Science and evolution was not there for having productivity for us, but that's what science has to do now. But what has happened during the evolution can unravel now, and we understand it step by step more and more. So this whole RNA world that you find nicely described by Czech and Stipes now, we have to see how we can use it, how we can take advantage of it for all the steps that we have to do in plant development. And one of the most effective is done now, the CRISPR-Cas system. Again, I will not explain it. It's a review article that you can see. Others every month come out now. It's known in the medical field and in all aspects of life science, how you can efficiently change in a way certain genes on the distances sometimes of 25, there are now more than 30 nucleotides, and bring them in new properties. So the in vivo biochemistry is now taking enormous steps, and it can be done even if a gene has many copies. So that means that now plants that our society so badly need that there is more work done, like wheat, where the genome is very big, sugarcane, where the genome is very big, that if you modify one of the genes, you can modify at the same time already the 10 or the 20 copies of the gene that is present. And I can assure you, this technique is slowly developing in our plant science group. 2 years ago, when I was in Shanghai, the principal investigator came from the University of California, but he just showed me the results of 17 genes in Arabidopsis that he had modified with one CRISPR-Cas, I call it transformation, presentation and reaction. So 17 copies were altered, and it was already 2 years ago. So there are countries where the technique moves, and especially for China, we will have to come back on that. So here I symbolize the approaches that I was mentioning in the beginning. There is enormous work with these new techniques that now are available, that you have to do in the discovery world. The best will always be in the public sector, because in the public sector, when you do the discovery, you can go on. And as was written in one of your seminar rooms, that chance is the basis of progress, that Francis Crick said. Well, it is, and in discovery, it's then up to you to say what way you will follow. But during your studies, it's sure that you will find 10, 20 questions that looks interesting. And try to get organized, and now with our European programs, find collaborators in your country and other places in Europe to go on with these questions, to run them down. But know that we also need to think about the applications. And applications, that the best is done by industry. Why? Because industry has this unique possibility of focusing. And it's, for scientific research, it's very often dramatic. If you are a scientist in a company, they will say, perfect, congratulations, we take a patent and we shelve it, and at the moment you will work from now on, next week on, you will work on something else. And what you find will be sold, will be transferred, or will be never used. And but it has to be done, the application, because if we go on, every week we find something else, and we say, oh, that's interesting, oh, that's more interesting. So many things will not progress. And that's an advantage of a competition in public research also. You can curse it because it makes a hard work, but you have to see. But also, and that will, I want in this talk, I'm only at the very first of the beginning of my talk. So have in mind that all what you do as application, and that's why I symbolize here hybrid canola, that I said that we did in the 80s, at the end of the 80s, and it's only used for canola in Canada with a high yield. People have really, when there are stress conditions, they have double yield, otherwise 30 or 40% more. It has been introduced and shown to work in India and in Bangladesh for mustard. I think Bangladesh will take it up because it's anyway off patent. So in principle, when it's off patent, you could think about generics, but the world of the multinationals is there, that they keep the generics also under control, thanks to the collaboration in spirit, at least, of some movements and non-governmental organizations that say that GMOs are dangerous, so that you have to control it. So that means that the big companies now can say, oh, there is a liability. If we give it to you, we are still responsible of the use you will do, so you cannot have it. It's not a patent that blocks, but it is all the work of the green movement. Well, some people did it with the best of intentions. That makes regulatory that the whole field is blocked. So for that reason, I use that innovation can be fantastic, but if society doesn't accept innovation, you are nowhere. And if science is complex, applications, especially in our plant world and in the life sciences, medicine it's also complicated. For all organisms it's complicated, but because of the need, because it's that I should have stressed when I gave the first two slides. Why are we thinking that these applications that we are doing feel special attention? That is, and we will come back to that, that is for the Sustainable Development Goals that the United Nations has proposed last December, that we absolutely have to progress, especially in the developing countries. But to do that, we have to do this. And talking to society is very, very complicated, especially since in our educational system, we never know about what did philosophers already think about humans and their society. You can use the word human ecology, but do you really look to humans as living organisms that make their societies and how their rules and how they feel of their societies and how do they do their decision making? Do you really learn about the economics of a human society and why public and enterprises you need both and what is the balance and how can all this makes the decision if you can go on with your science. And that is what we have to think about today. Because all these findings that I mentioned, you have seen it often. It's only some impact traits that are used, the BT, the hybrid, the herbicide resistance, and for a very limited number of crops. It's of course, the BT is done for inside the plant, for the roots. So there is a lot of different progress, but it's still, if you look for the applications of GM technology, you can say it's 10% of the agriculture in the world, because it's 180 million hectares. But it is the large-scale agriculture that is controlled by 5 or 6 companies on the maximum. And all what really the developing countries need there, a lot of progress is there in the laboratories. And lot of other applications that using agricultural waste as a source for the chemical industry, since we know we are running out of petroleum. We have to, instead of burning, learn to use all these fantastic molecules that have been made in plants. And it would be possible. And we know about fermentation and our progress about microorganisms, bacteria, yeast, some of the fungi. There are so many organisms that we know how to use for ferment. Well, all the city waste, the agricultural waste, we could use it. We know we can progress because the metagenomics, we can analyze and find a lot of enzymes, but really making the optimal organism for doing this fermentation, you should not believe that you will find it in nature. Why would nature make a bacteria that is optimum for our overpopulated world to have less pollution? You have to make it, you have to learn to make it, and you have to explain to your colleagues in environmental studies that we have all the tools to do what they think that is needed in our planet, a concern for the pollution that we are creating by being so many. Best we do bird control and make it that overpopulation goes down, but that's another communication problem with the society. But it could be done, but at least at the moment, most urgently, focus the applied science on finding these types of solutions. And it can be done because you follow the literature very well, you know fingers that are less adapted to small buttons. So plant biotechnology has given so many possibilities to make to solve problems for health through adapting that the plants have a medical value or have a better nutritional value, because nutrition is medicine. We have plants that are better prepared for the industry. We have plants that can overcome all the possible stresses in, not brilliantly, but the first steps are there. And with the systems biology and with the RNA world we are discovering, we will improve it. But already those could be used, and they are blocked. There are applications in stress that are worldwide to really explain to society. They are not complicated to understand. You know, in Europe, the potatoes are attacked by a mold, there's the blight, the Phytophthora infections. It were dramas in the 19th century. Millions of people from Europe, especially from Ireland, but from the Belgium, north of France, Central Europe, when the potato fields were destroyed, there were no chemicals for giving protection. There was starvation, and people were like the immigrants that we now see coming in Europe. That was the European that were moving then to the States or Australia. So making a plant resistant traditionally is done through breeding. Some of the people who believe in that everything is already there in nature and that a chemist should not touch an organism, they believe that only breeding will bring the progress. So people tried. Already in the 50s they tried, because in the Andes there is a potato that is resistant. So it was So they did the breeding. Okay. Now I'm sitting in the bananas. They did the breeding, but it took 35 years, and they obtained what's called toluca, and it is indeed mold-resistant fruit breeding, but it lost so much properties that nobody wants to eat it. It's a horrible potato. It's good for industry to make starch. For starch, often potatoes are used for making starch. While for gene engineering, in 10 years, and then most of these years are still used for the regulatory, you could stack a whole set of genes and have the double, triple resistant potato with a whole set of genes. In the States, this has already been approved for production. And the work there was done by Jonathan Jones in the UK. And he started up a company in the States because he gave up on Europe for that. We still believe that we will do it in Flanders, that Flanders will be better than the rest of Europe. We'll see. The Dutch already think that maybe they should not use it. They have done it, but although they badly need it, that's the problem. We can discuss it later, but on this Phytophthora. Transgenic bananas. We have all the collections of bananas in the world are in Leuven. We want to have improved bananas because there is a lot of like Black Cicatoga, a lot of diseases caused by fungi. There are a lot of antifungal peptides known. We can make in the laboratory transgenic bananas. We could already convince Uganda to have 4 or 5 of these lines in the field, but Uganda is a very complicated government. We never know if somebody with money comes in and convinces them that they have to close down. That will happen next year. It happened in Kenya, where everybody was thought ready to go because Kenya badly needs also these bananas. Well, we'll see in Africa, it's a race because the realizations are there and people see it, the farmers want it. They cannot understand why they think it's just Western bad will not to give them, to have solutions for their problems and not want to give them. For them it's difficult to understand this communication with society aspect. The Bangladesh example on eggplant is fantastic. In India, there's about 500,000 hectares of brinjal, of eggplant. People, this plant is very easily attacked by insects. The BT brings solutions, but whole India for 10 years now goes on spreading a chemical that is highly toxic. It's a neurotoxin, it's a phosphoramidate. Many children die every year, many people go sick. The farmers know it, the farmers hate the chemical. India produces it. In the old days it was Bayer who produced it, but it has been forbidden by the Convention of Stockholm. India has not signed the Convention of Stockholm, and it is a lot of, in Bhopal and a lot of chemical industry makes it. So at the moment it's not accepted. The lady who is now Minister of Agriculture in Bangladesh has said, we need it. And she convinced well, it comes from Monsanto, the solution, and it is made by, the seeds are made by an Indian company in India, that it be used, and now since 2 years, all farmers adopt it, and the BT, brinjal, finally is there in Bangladesh. A place where nobody would thought that the transgenic plants would come, because there is somebody in the government who sees how important it is and makes it possible, and the farmers immediately spread it. And we know already that the farmers bring it in clandestinely to India, well, Pakistan, since they both are Muslim countries, they are very interested to start using it. So we'll see how that evolves. Another application that is easy to understand that will be important is a former student, Luis Herrera Estrella, and one of his senior scientists who did it, and you can see the work in Nature Biotechnology. We have a problem in intensive agriculture. We need fertilizers, phosphate as fertilizers. But only a small fraction of the phosphate is taken up by the plant because people want high level of phosphate to see good effect. Most goes in the rivers, makes that in the rivers and in all the coastal waters, all the microalgae, the diatomate bloom, because there's suddenly so much phosphate. At that moment, with all this phosphate, if this diatomate are there, then they use a lot of oxygen. Then all these waters back in the Baltic Sea, along the coast of France and the UK, in the Gulf of Mexico and the States, become atrophic. The fishes die because there is no oxygen, because of the intensive agriculture we do. But we need it, this intensive agriculture, because our societies are made of it. Possibility that is worked out by the two there is using phosphite. There is a lot of phosphite in the world. It's there. It's not used because phosphite is not taken up. So what you can do is engineer what has been done for quite some model plants, that the phosphite is transformed inside the plant, is taken up and transformed as phosphate. So if you then don't give phosphate fertilizer, but phosphite fertilizer, then at the same time you have a herbicide. You don't kill, but by just not giving phosphate. So the weeds cannot grow. So your crop grows, and the weeds don't grow. So you don't need to have a herbicide. This system works well. So let's hope that nobody will find a negative argument for that, because at the moment, after several years of testing, nobody has found something negative. So let's hope that it can go around. Give this here as an example, because in all the health shops you hear talking about omega-3, and you know that it's healthy, this omega-3 fatty acids. But a lot of plants don't make it. So you can take the genes from the plants that make omega-3 and put it in other crops. That has been done in Rottenstedt. And that's only an example of making more nutritious plants. Ingo Potricus, 10 years ago, in 1999, he showed the way to make provitamin A. That's very important in developing countries, because there are millions of children that become blind, and many that die because of shortage of vitamin A. That is overcome, but nobody gives authorization to use it. Meanwhile, the same consortium with Ingo as a driving force has obtained that at IRI, the International Rice Research Institute, at the time that there was a director that was interested in transgenics, that they could make rice that has more iron and more zinc. And deficiency of iron and zinc is also very important in developing countries. And recently in Ghent, Dominique van der Strade, there was a way to have folate in rice. Everybody knows about, at least women who are become pregnant know that the doctor will say folate is important, otherwise there is a risk of spina bifida, there is a lot of developmental, because there is not enough folate in our food. But we can, at the Social Security, can pay for additional folate. But in the developing world, it doesn't exist. So now we could stack in rice, vitamin A, folate, zinc, iron, it's there. But nobody wants to go for it, and nobody gives authorization. So that is quite an aspect if you think about what is plant science. Another story, the one again, of course, I take a lot of stories that were done also in Ghent. That's the trees, the poplar trees that used a lot in our countries, not only for wood but also for pulp, for paper production. So you have to extract and separate cellulose, lignin. That's a process that in the old days was done with a lot of sodium hydroxide and chloride, very polluting industry. Here you can show that with just one change in the lignin, by the methylation of a phenolic group in the lignin, that then at the moment you have double yield in the extraction of cellulose. That's the function of time, so you can use much less polluting conditions for doing this production. You can stop and have a high yield here instead of going on with this type of treatment. Could be used at the same time. We can probably, with the same approaches, make hard wood from soft wood. That's also very important for some examples where you would use wood directly for burning. So that are other applications. We heard about the Ebola story, but few people know, it was an article in Nature Biotechnology, that the first antibodies against Ebola were made by techniques worked out in Ghent and were done in plants. And there is now an enormous interest because we know there will be many diseases, new infectious diseases, and the Zika virus is already there and others will follow. If we are so overpopulated and the way hygiene is in all the slums and all over the world, we know that there will be a lot of other disease. Each time we will have urgently new antibody production, but if that has to be done in batteries of animal cells that cost €4 million for building, you know that it will be slow. And also there is, also in the pharmaceutical sector, there is a problem of monopolies of pharmaceutical companies. So there is a real demand of making in an efficiently way proteins in plants. It can be done. And so we will see if it will be done. I have to accelerate, sorry. We are now with the microsequencing at such a low price, and by understanding also the RNA world and doing the RNA sequencing, studying better and better all the organisms that are in the soil. And we know that each plant is surrounded all over the plant and surely in the soil by organisms that are beneficial, and that a lot of diseases and problems become through the mishandling or not availability of the positive organisms for the plant itself. In the medical field, progress was tremendous in the last years to show that we have actually, that each person has maybe 2 trillion cells, but that he has 3.5 trillion of bacteria in all his orifices, and that these bacteria are there for making compounds. And if you do biochemistry, that you don't have to look to the biosynthesis in all the human cells, but also in the microorganisms, because they make a lot of important compounds for humans. It's exactly the same for the plants. We knew about nitrogen fixation, but we don't know about the free-living nitrogen fixation that are inside the plants, the endophytes, that do a lot of delivery of nitrogen and a lot of other compounds that are there, and the protection against the pathogens. So this holobiont, as it's called, this studying at the same time the plant and the microorganism, thanks to the new tools we have, is really very promising. And it is important that it happens, because we have all these orphan crops that till now nobody worked on. And out of here we could have a blooming and interesting plant molecular biology by doing the fundamental research and have it applied, and bring services to these countries by setting up mixed startup companies where the science is done here temporarily, and they rapidly will take over. We have seen how well Vietnam does, how well some remote parts of China are already doing in 10, 20 years. Here we could, if we do it on purpose, with our enlightened democratic societies, it would be very efficient if the will was there, and if the other reasons are there, then we can close the communication. We will not go into detail, but just I want to attract attention of one that is really probably not known, Datirus sativa, called grass pea. It isn't the little peas it makes, but it's a plant that's extremely drought resistant. That's an example in Ethiopia, when there is drought and even sisal chickpea is becoming yellow, Datirus is still deep green. And in Bangladesh, if there has been a flood, 2 weeks, 3 weeks of water, all the plants disappear till then. The water goes away, and immediately Datirus grows. So it is very resistant against drought, against anoxia. So there is, for fundamental research, it's fantastic. But there is a toxic compound in it, but only toxic for humans. But by itself interesting, because it's toxic only for the nerve cells of the lower limbs. Till now, no neurologist make a distinction between the nerve cells of the upper limbs and the lower limbs. So already this toxic compound is by itself interesting. So knowledge that's around for 30 years that we tried to promote, but never could find funding for it, because oh, you will be there with your GMOs, and nobody wants these GMOs, so don't start working with it. So again, we have shown in the talk how important it is not only to have the discoveries, but see that innovation can work. So we have to say some more word on this problem of communication. We have to explain to society that with this the Sustainable Development Goals that the United Nations propose, extension of the Millennium Goals, that indeed it's absolutely essential that we end poverty. And that poverty means half of the world at the moment, because half of the world has less than €2 a day. And that we know very well our society. We know that we need to have a just government, and we have fulfillment of human rights, and all this is extremely important. But if we had peace and security, it would be even better. But that is only possible if, and there is the circle round, if the poverty would not be there, because otherwise if the poverty is there, then people will revolt and will not bring peace and security. So we, with our science and technology, we have to see what can we do now as innovation that can already help, and that slowly will make that society wakes up and takes care of these different points. And that, I think, is a responsibility. Because sustainable identification, the green people don't like it. They say, oh, Norman Belloc with his green revolution, he is responsible of all these chemicals that have been used and all this pollution that agriculture is doing. Well, it's not Norman Belloc, it's the overpopulation who did it. overpopulation needed food, and nobody was able to communicate birth control because when people are poor and they have no social security, they need to have 10, 15 children because many will die in between, and when they are old, only some will help them, and the state will not help them because there is no social program to do that. And that is what has never brought the whole picture, that if we want sustainable identification, that GM technology can help to it, but that we will go on at the moment with our situation to have on one hand intensive agriculture, what people say, the big landowners that exploit and that pollute the soil. You cannot without that. If you bring in a lot of new crops, and if you bring through science that in these areas agriculture brings more. We have seen in Burkina Faso, just with the BT cotton, if the income doubled, the first reaction was that we send children to a school where they learn, because they normally go to the school where they are with 45 children in one room, and children from all possible ages. There is private schools where the children will learn, but you have to pay. And then the people say, if we go children to the private school so that they can do maybe secondary school, they cannot do it for 15 children. So they normally think that maybe 2 or 3 that will do well and go secondary school and will become in administration. If it is a myth or not, I don't say that is the rule, but it looks not unlogic, and we have to see, well, as scientists, to discuss it and see that we can help promoting it. I want first to say that trees were absolutely blocked by Greenpeace because the pollen flies too far. And surely, since GMO are dangerous, pollen of transgenic trees must be very dangerous. It's just what inhabits in nature and all the processes that go on, and we should realize that there are not... Is it the alarm clock? Well, then I rush. The transgenic trees have the authorization of the Brazilian government, but the industry is still very, very puzzled. Will they start using them? Will they start fighting against Greenpeace, or will we just see if there are other ways to escape and still shelve a bit the transgenic trees? But that's for discussion. At the moment, if we now talk really about what is GMOs, stress that there is, after all these 25 years that people have been using it, there is not the slightest argument of any danger that ever has been seen, notwithstanding Mr. Serralini and some others that make articles. And also for the environment, no scenario was worse than the normal agriculture we do. On the contrary, many points for solution are already there, and many types of agriculture that are better can be done. Is it unnatural? It's the basis of evolution. And so if you see the tree of life, you see all the pieces of DNA that were there some 100,000 years, 100 million years, 800 million years, a billion years, that are still used at the moment in another context, that have moved around. Finally, for people who were doubting about if evolution existed, now all the tools of sequencing, every week, there are new articles to show that humans have genes from Neanderthals, that humans have genes for other, Homo community that has completely disappeared meanwhile and doesn't exist anymore. We see all the links, how it is built up, and whole evolution became a fantastic science. But most of all, it's clear now that nature is one big genetic laboratory. And you observe it, and that's all what you can do. And there is no purpose in it. There is the things that happens in nature. But you can analyze, and you start having these tools. And so it is absolutely misleading to say that there are human genes and pig genes and rat genes, and that of course if you are a pig gene that you put in a plant, if you are a Muslim you are not allowed to eat this plant. If a human gene should not use it because you will eat your ancestors. All the genes are our ancestors. But that's what told, and people believe it sincerely because they've never been exposed to science, so you can tell them all kinds of stories. And there, slowly, we are not, unfortunately, there. An example, very recently, the group in Ghent, together with SIP, could show that sweet potatoes has, for thousands and thousands of years, the whole tDNA. And that it was probably an advantage for conquer new species, and that people then had attention, and all the cultivated, all possible sweet potatoes from all over the world have been analyzed, and they all have the tDNA. So it's at least 4,000 or 5,000 years, because agriculture, you think, is 10,000 or 11,000 years. If now somebody from Greenpeace comes and says, your experiment has not been done long enough. Sorry, it's only 5,000 years. Sure, it can be done longer. If humans are still around, with all the dramas that will meanwhile happen. So that we have to see. Even more, if you still doubt about horizontal gene transfer, look to that article in Nature in 2014, that upon grafting, or in nature, when trees sometimes grow together, there is an enormous pieces of chromosomes that are exchanged. There are, through grafting, people have made really new species of plants. That was in the Solanaceae, like grafting tomato to some of the other Nicotiana species. So horizontal gene transfer happens a lot, but of course we could never see it. But now with sequencing we are seeing it, so we have to tell it. Horizontal gene sequencing is the basis of evolution. There is, of course, the fertility, but once more, in your lecture room, you have a nice quote of Darwin, that it really the species that make it, that's the ones that are the most flexible. And the DNA molecule, it's fantastic, you have to think about it. It's an example of something that is stable on one hand, but has a maximum of flexibility. And it has always been compromised on the data that have been achieved, and that were for a certain environment, certain living conditions, and then being ready to adapt if there are stress situations, if things go wrong, if there are climate change, if there is drought, if there is all kind of interference, if there are humans coming and spoiling everything. So that is how plants and all the organisms survive. And that's the DNA. And that is something that we should learn to communicate. And if people say now, as a big discovery, oh, the precautionary principle. Everywhere you hear about the precautionary principle. Actually, it was a term coined in marine, how you call it, insurance. So the insurance, when are you sure that and what have you to do to calculate all the possible dangers? And then it was extended, and I thought it was nice to find that already in 17th century it was said that you have to consider only the gravity of a hazard. So you have to calculate the probability that it will strike. Because you know the human mind. All the things where you can take an insurance, against the monster of Loch Ness. Have an artist in Brussels who has done it. In those days, in the 60s, it was cheap, just paper handling. But you are protected and proudly in your office you can show, I have an insurance. Everybody will agree that the dangers for being hit by the monster of Loch Ness is... But there are many things that people, if they don't know the science behind it, and it's not for laughing, it's now really dramatic. It's the problem of communicating science, that you can make them believe things. There are many, many things already. Voltaire has said that if you make somebody believe things that doesn't exist, what then? If such a thing is possible, if we live in such a society, then you will end up with people that will do the most atrocities. And that was already the wisdom that was around. And there I come to the final slides, that we have to do what is thinking. And if you want, if you are interested, Daniel Kahneman, Nobel Prize of Economics, it's a 600-page book, but really it's worthwhile. Each time when you think you have it right, he will prove that you were wrong. So we are all sloppy thinkers. We are all lazy in a certain way. At least we want always the easy way out. And that's where we fool ourselves so much. And that's the polite way. Another Nobel Prize of Economics, he really studied the human emotions, how they are doing the decisions in stock market games that make other people rich. Then see that can be analyzed, but discussed. There is no solution for it, but at least be aware of it. So this whole situation, we tried to analyze a bit with our Department of Philosophy, and that's an article in Trends in Plant Science that some of you may have seen. So it's the story, if we really think about essentialism, that if you tell to people about tomato genes, that's really the essential of tomatoes. So if you touch it, you will spoil the tomato, or if you will bring it somewhere, if you bring a new fish gene in it, the tomato will smell fish, because another problem that we see if we start analyzing the reasoning society often has, and I do these superficial things because that's what often has been called the theological trial, that if in evolution something evolves and then another thing is built up, and after a time you see like a line, and it seems, oh, it's fantastic. And then of course creationists can say, oh, it's really intelligent design, how evolution built up, and it's intelligent design. But if you do DNA analysis, you see that evolution is the worst tinkering is possible. It's just like somebody who doesn't know the job and has been making all kind of, a lot of dramas are there that could be avoided, and a lot of things interfere and block other types of development, because that's the way evolution has worked. So there is no line, but it looks like that if you have to build on something that was existing and then you build on model. It seems like there is a line. And then there's the whole story of disgust. It's very complicated, but we all are disgusted by one or other thing that we don't like and we think is, sometimes we use the word unethical, or society cannot accept that. People are disgusted for some aspects of food. But we have seen in France that millions of people were demonstrating against gay marriage. What is the rationality? Why would you be against? Only thing is, I'm disgusted, I cannot see it. So if people hear all these stories about GMO, be aware. Some people will say, I'm disgusted about GMO. I never want to eat any GMO. Know that it exists. And that's only the very, very, very first steps to think what is really going on in nature. I took as a symbol this painting of Magritte from 1933, La Condition Humaine. I call it the demanding ethics of knowledge. Really, we have some knowledge, and we know that all the effort we do in our laboratories, in our scientific research, to improve the knowledge and to do it better. And always ask questions. And that's important. And that's a real human endeavor that we can try to explain to everybody. That's what has to be done. Go for knowledge, try, challenge knowledge, ask what it is. And that has often been blocked for social science, economic science. They have worked in their own world. We should also realize that, I call it the joys of fundamental science that we are doing, are very, very recent. When I was a student, I had in my textbooks that DNA is for animals and RNA is for bacteria, yeast, and fungi. And that was it. So there are, and if you go back to the early 19th century, what you can feel, and all the people that made what we call philosophy, our thinking about society, our feelings, they have been influenced, and they really were not so keen on knowledge. But it's very understandable. People should analyze what are humans, what are the pleasure, and that's why I took a painting. We are a pleasure in art, the way we enjoy, but that's human life. And the rationalists in research, the rationalists in engineering, if they are not pointed out that they don't know what a human is, that they don't know what society is, and then they want that humans would accept this knowledge of society. You can understand that it doesn't work. So I was pleased to hear that you have social science also present together with the development in agriculture and the development in fundamental science. In Paris at the moment, Paris, the new university they made, they also have not faculties, but inside the group of molecular biology, they want to have people that relate to the faculty of science, that with you can talk about philosophy, about society, about all what moves people, what makes the pleasure of life, and why to see if we live in such a society with so many billion people, with all the problems, we cannot say we go back to the Garden of Eden, we don't want this type of world, we don't want this type of pollution. We have made it, we are responsible to solve the problem. And science and technology can solve it. Because I think the word demanding ethics, because it's very difficult to do it, it's already demanding to do the science, and really to see the science in society context, ethics really important. So I took another one from Salvatore Dali. It's called Premonition of Civil War from 1936. Made, he says, from boiled beans. So we see the danger that we hear every day in what's happening in Africa, what's happening in the Middle East. We know it's the society who's enabled to bring solutions. We can see with our science if we can. I am convinced that in the 21st century we will be able to progress with plant science. It would be summable absurdity if we not. But everybody has to do an effort. Everybody has to be involved to explain. And if he has his doubts, that's absolutely normal, because we should in the first place realize that as scientists or non-scientists, we are all the same. Ask the questions, work for it, and then talk in principle to the green militants, because they know at least the importance of nature, show them that we are all allies. And it's only when we will do that, that we will have in a better environment the little bit of tropical forest that still exists, and then we will see, hopefully, that Greenpeace makes manifestations against the burning of all the forest that's still left on some of the islands in Indonesia. Now Borneo also has done. Malaysia was already out. So, and more is threatening us. Let's unite for saving that, and that will be for science and technology. Thank you.
Thank you very much. No, don't worry. Thank you very much, Prof. Van Montagu, for this outstanding talk. So we have now time for questions. We are a little bit behind schedule, but I think we can look for some questions.
Otherwise,
well, maybe I can start by asking you, when you discovered the TI plasmid, well, at that time, when did you realize that it would be a breakthrough for the developing of plant sciences in the next years? You immediately realized that, or
no, we even didn't think about it, because we all were close to microbiology. So we were so taken by pieces of DNA that moves around through organisms, and now it moves to plant cells. First, well, we had during the World Food Prize nice discussions with Marindel Chilton, that she went to Agrobacterium because she was thinking that we were telling uttermost nonsense, that it was possible. Because other people, like always, had already shown that there would be, if you just add DNA, it could be taken up. And I think it will, even that exists, but at that moment, people wanted already to do gene engineering with it. That was around. So she tried to prove that we were sloppy workers. So then she saw that we were right, but nobody had the tools. Southern blot didn't exist. She did it with cut curves. And we thought it's too much, it's too hard work in cut curves to work during 24 hours. And no, that's not possible. But she did it, and then together we were convinced with the opine stories that you could have the same strain that gives noparine as an opine, and then there was another strain that gives octopine as an opine. So it was the bacteria who determines the properties of the plant cell and of the regenerated plant, if it was a teratoma plant, because we didn't have normal plants. So we, at that moment, we thought that's the most logical. So let's go to find out the details, what is needed for doing the transfer and what can be removed, and then it was immediately erased, and then we believed it, but it's a growing process.
Yeah.
In the beginning, we knew it was extra-chromosomal, but it could be a virus, it could be an organism that we didn't know yet, a mycoplasma, because they were not so well studied in these days. Yeah? Okay.
Sorry. First, thank you so much for your excellent presentation. You just realized, giving your hope that the progress will be towards the use of GM plants. But do you think that this will be based on political decisions more than consumer acceptation? Do you think that the governments should just consider the science-based decisions, and for that we need a lot more activist groups that try to mobilize the people for opinions against it.
I think everybody has to work in the same direction. Who will have the most influence, we surely don't know. In last point, it's the product that will count. If there is something that people need, people will use it. So, cotton that you don't have to spray and that will be resistant to the insects that attack it is there to stay. And the large cultures will follow. Now, it's only the major companies who will do it. I think with the new breeding techniques, and if people, some of the possibilities that are around, really push hard enough that the product is seen, that will also already change. If China, who spent $45 billion for buying Syngenta, they will apply it. It's just to have food quality extension services, to have a real good agriculture service that you cannot build up, that they have understood that they have to bring in, and in that way they have it. So it will be there. So they will use it. But at that moment, if they start using it for orphan crops, then Europe and the States, China works very close with Russia. Just like China, Russia absolutely blocks GMOs at the moment. But together, if they start developing it. So, what can I, no crystal ball, only in science, there is no argument against it, there is a bad need for it, there is a lot of possibilities, and there will become more. So, let's go all together again. Anyway, the science we find is excellent, and you will find a lot of interesting stories out of that. So we have to go on and talk to society to convince them. And in that way, GMOs would be very spectacular if they could wake up people, because we have so many beliefs that things are not good in technology, and there are reasons the way we apply it. But in a more conscious way, applying some of the technologies would be wonderful. Look to the programs of a lot of the green parties. There are a lot of needs where science and technology come in, and hopefully then GMOs will have shown the way. But that's wishful thinking. That's another belief. Yeah.
Professor, this was such a beautiful presentation that is really challenging to try to ask you something. But what I'm interested in hearing you is about the speed of knowledge accumulation, and try to reconcile this speed and this accumulation with the fact that you have most people not in contact with science, and now how can we make them understand that this knowledge can be useful, in fact, and when it is applied, most of the time it is applied on the human needs.
I still think not abandoning it. By example, I saw it 3 weeks ago in Brussels. Seralini was again there with a story of something dangerous that was going on, and now pushing around glyphosate and so on, the danger zone. And at that moment, the Member of Parliament of Greenpeace, José Bové from France, was there, and he even anticipated and said, yes, we are not against GMOs. GMOs are used in all your medicines. GMOs are used in fermentation, in industry, in cheese. What we always use as argument before, that in the days of the mad cow disease, it would be dramatic if there was not the enzyme through genetic engineering that was already available, and that we didn't have to use the stomachs of the veal like it was before for making cheese. That all the cheese is made with genetic engineered microorganisms, like a lot of beer, but don't tell it, beer is. There are some of these people who dare to touch beer. No, all this is there, but Greenpeace was already saying it. Is it ready for the escape if they have to give up? Or is it just another way to say, but food is sacred, nature is sacred, and you cannot do it to nature because if you spread, it's always the argument, the genes will spread in the wild. The mystery of the genes that will spread in the wild. The facts are there. So if people hear that, if we help them to make conclusions, for other sciences, like for radioactivity, it's complicated too, because nobody knows what to do with the waste. But for the radiations at the moment and the engineering systems, it's less polluting than if the CO2 emissions, and especially the NO emissions and the NO2 emissions that are going on from a lot of industry. If that is done in a honest way, Claudine?
Marc, what I really enjoyed in your talk is the way you went from fundamental research and the importance of the fundamental research to the application. But my question about GMO, because since a long time I'm listening in several meetings, voices against GMO, etc. Not long ago, I heard that GMOs influence deeply the ecology. So I would like to hear from you, because you have the experience, if there is any modification of the ecology, of the ecological systems which are surrounding the GMO plants or GMO products, GMO. So I would like to listen from you if this is really true or if it's just a fantasy.
No, it's absolutely, it cannot be the mere fact of being a GMO, because all plants through the breeding is new types of DNA that are brought in. It's not a technology. It could be some of the products that are used, but glyphosate, whatever people say against Monsanto, glyphosate is one of the herbicides that are degraded the best in the soil compared to all the other herbicides. Do we need to use herbicides? People say, no, we can. They have been, the early ones that were used in the 60s and early 70s, they were really dangerous. Now they are, most of the herbicides are much less, but everybody in toxicology knows that it is the level of the doses. Even water can be toxic in enormous amounts. So we have to see that there is a diversity of herbicides that are used, and if we can avoid it, because they all make a lot of collateral damage. It's not that we eat them, and then it would not be damaged. Herbicides make a lot of collateral damage to the whole flora of the soil, because they are like, in a certain way, like antibiotics. It's very often, it's a bacteria, basta, We came to basta because there are bacteria that make it to fight other bacteria. It was really like an antibiotic that you could make in enormous amounts. And that is damaging, surely, for environment. But environment is a permanent warfare. Nature is a permanent warfare. And that we also have to be able to communicate. It's not only GMOs we are able to communicate. What is really nature? I think humans must understand that it is an important question and it deserves to be considered. We should push that. The same for evolution. All things where we are really sure that science knows what it's telling. The details change, or certain visions change, but the fact, it's not that the facts were wrong, but their interpretation of the facts was sometimes a bit limited. Please.
Well, thank you very much for your very interesting talk. I just would like to give you a very good news. You talked about the importance of underused crops and to work with underused crops like Latius sativus. And indeed, here at ITQB, we are working on Latius sativus already for a long, long time.
Okay.
And I completely agree with you that it's extremely difficult to find funding to work with these plants. But indeed, they, especially for instance like Latius, they are so resistant to several stresses that they can be an answer to many of the modern agricultural problems. But the good news is that the Portuguese government decided to funding a project that is starting this year on this particular species. So we hope we will be able to contribute to this too.
Thank you. Please send an email. We still edit Latius newsletter, but all customers are in Bangladesh and in India and Ethiopia, so we can talk about science.
Yes, exactly. Well, Professor Montagu, just I know that your institute is very active on Latius sativus through Professor Langwijn. So we are keeping in touch with him.
Okay, excellent, thank you.
Okay, unless there is a burning question to finish. No? No further questions? So
There is one in the back.
just to finish, really
burning question, Professor Van Montagu. Do you think that if you had called GMOs otherwise, the problem of having so many people against it would be solved? Do you think the fact that you called it genetically modified organisms can be the problem?
It's an evolution. The word is, of course, very important. That's why clever people have taken care that the CRISPR-Cas, it's called new breeding techniques. So surely people are sensitive to work, but it's only because there are so many words that you could be shocked by that are around and that are used in daily language because nobody did actually name. So it's really because there was let's say a conspiration, although we don't like the concept of that something is a conspiration against, but sometimes in our society, sometimes it exists.
Okay, so thank you very much, Professor Marc, for this excellent talk and discussion.